Today we will discuss about whether the pancreatic cancer cells that express the different markers, that is CA19-9 and sTRAs, belongs to some different group of phenotypes or not. Uh, while we have touched upon this topic in one of our previous video, in this video we will discuss this topic in slightly more detail. So what is the final outcome that researchers has find out is that dual label cells are mostly represents secretory epithelia, whereas single label cells are mostly represents this dysplasia environment, or their tissues are seems to be more damaged and they are poorly differentiated, whereas dual label cells are kind of cohesive and they are well differentiated cells. We will discuss that results in this video. So we will start with this H&E staining of this MUC5AC. H&E staining through this, you can say that sTRAs and CA19 Glycans is shown on the right hand side, whereas MUC5AC and beta catenin shown on the right is on the right hand side, and this sTRAs and CA19 Glycans is on the left hand side. So what we can see that in the well differentiated cells, we can see the presence of both MUC5AC and beta catenin, and that can be seen, and at the same time these sTRAs and CA19 Glycans, both of them are also expressed. Whereas in the case where the differentiation is not proper, we cannot see this MUC5AC's presence. However, we can see that beta catenins are present, and same things we have observed for this and another case where the differentiation is not proper, where the C99 are more dominant compared to the sTRAs. And here also we see that beta catenin's presence is there, but MUC5ACs are not present, indicating that MUC5ACs are more predominantly present in the case where in the case where cells are well differentiated. And in some of the other examples, we can see that none of them are showing any presence. That is, MUC5AC and beta catenin are not present, whereas there is sTRAs and C99 presence is shown. And this case is this is the case where the cells are poorly differentiated. So this indicates that as the cells are less differentiated, they will have less MUC5AC and beta catenin. In the moderate differentiated cells, that is second and third, we can see that no MUC5AC or very low MUC5AC is present, whereas when the differentiation goes further down, we can see that the beta catenin presence will also goes down. And in the case of CA19-9, in the well differentiated cells, we can see that there are no presence of MUC5AC and beta catenin, indicating when only CA19 are present in the well differentiated cells, they do not shows MUC5AC and beta catenin. However, in the well differentiated or moderately differentiated cells where sTRAs are present, it shows both MUC5AC and beta catenin, indicating this sTRAs are usually more predominantly found in the well differentiated cells where MUC5AC and beta catenins are also present. And thereafter they check this overlay of MUC5 and beta catenin in a dual expressing well differentiated foamy cytoplasm. On the cytoplasms they checked. So when the cytoplasms are clearly expressed, clearly visible, they can see the presence of both MUC5AC and beta catenin. However, in the first case, the expressions of these sTRAs and CA19 both are present. However, the overlap regions are less. And in the second case, we can see that the CA19 expression levels are high and sTRAs expression levels are also high compared to the first case. And in this case also the overlap regions are less, and we can say that this is moderately differentiate cytoplasm. So when the cytoplasm differentiation goes down, we can see that decrease in the amount of MUC5AC. And when in the third case where there is no overlap of CA19 and sTRAs, but both of them are present independently, we can see the presence of both MUC5AC and beta catenin. And this is also about the well differentiated cells, but it is not covered by any stroma, foamy cytoplasms. This is well differentiated foamy cytoplasms, and there is no presence of any stroma, whereas in the first two cases we can see the presence of stroma around the cells. And in the next case, in the vacuole or poorly differentiated cells, when in the first examples the C99 is dominant. So from the previous results we can made a point that MUC5AC's presence will be lower and catenin 9 presence should be higher compared to the MUC5AC, and this is what they have observed. And same things are true for the second case, where also we can see that in the poorly differentiated cells the CA19 and sTRAs both are present, but there is very less overlap regions, and this because and in terms of these proteins, the MUC5AC's presence is very low compared to the beta catenin, and when only sTRAs are present in the poorly differentiated vacuoles, we do not see any presence of MUC5AC or beta catenin, or very low presence of MUC5AC and beta catenin. So all this indicates that different criteria will cause different phenotype of the proteins expressed on the cell surface, and that is related to different ways to these markers, that is CA19 or sTRAs, and some of these phenotypes are discussed in this figure. If you need to revise or understand these phenotypes, again you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.